As RNA polymerase travels down a short section of DNA called a gene, it transcribes that DNA into an mRNA copy. But if the DNA has a mistake in it, then the mRNA gets a mistake in it and creates a protein that could be good, that could be bad, or just doesn't work. And those changes in DNA are called mutations. So by the end of this PowerPoint, you should be able to distinguish between chromosomal and gene mutations, and list and give some examples of different types of mutations. Mutations are generally categorized in, or they're put into two groups. The first type of mutation is a gene mutation, and that's a mutation that results to a change in just one gene that codes for one protein. So an example of a gene mutation would be in sickle cell anemia. There is a protein on your red blood cells that carry oxygen around, and if this protein gets mutated, then it could cause sickle cell anemia. And so people with sickle cell anemia have red blood cells that are sort of shaped like half moons. And that causes a whole lot of problems for them, including blood clots and anemia, which means that your cells don't get enough oxygen. Chromosomal mutations are usually more serious. It's a mutation to an entire chromosome or even a number of chromosomes. So a lot of chromosomal mutations we don't know very much about, just because chromosomal mutations can kill a baby even before his or her mother knows they're pregnant. Some chromosomal mutations are less serious, like Down syndrome, and so babies are born with Down syndrome all the time, and so we can study some chromosomal mutations that way. But you can tell chromosomal mutations affect the entire body. They'll affect different body systems, not just one particular process. So in Down syndrome babies have very particular facial features that identify them. They also have problems in their digestion. They've got problems with their heart. So a lot of different problems found throughout the body with Down syndrome, a chromosomal mutation. A point mutation is a type of gene mutation where just one nitrogen base changes. And sickle cell anemia is an example of a situation with a point mutation. So in this particular point mutation, we've highlighted the mutated codon in blue, and a T changes to an A in the DNA, causing an mRNA to have a U instead of an A. So this codes for an entirely new amino acid. Glutamic acid is what should be there, and valine is what happens, is the amino acid that gets translated if there is sickle cell anemia. So where does it come from? Mutagens are things that cause changes in a person's DNA. And there's mutagens all over the place that sometimes we don't even know we're being exposed to. And we're exposed to on a regular basis. But UV light is a mutagen that can cause cancer. Same with X-rays, and chromium-4 is a type of chemical. Cigarette smoke is one that I didn't include, but that's another mutagen that causes genetic diseases. And genetic diseases are just ones that can get passed down from the mother to a child. So in sickle cell anemia, if a person gets the mutation for sickle cell anemia, they're kind of the person that starts it out for their family, and then they can pass that mutation on to their children, and it can so it can have a place in the family that it just starts. So what's actually getting mutated is the DNA. And then that DNA gets transcribed. So RNA polymerase comes through and creates that short section of mRNA. And the mRNA then goes out to the ribosome, and it's translated into a protein that is incorrect as well. So there's three different types of point mutations, which are very well illustrated by this graphic over on the side. So the first type of mutation is substitution, and it's a lot like what it sounds like. You substitute one base for another one. And so in red here, it shows you the amino acid that ends up being affected. In a substitution mutation, it's just one amino acid. And so this is exactly what happens with sickle cell anemia. It's just one amino acid that gets changed that causes sickle cell anemia. An insertion means that a base gets added. So if a base gets added, what happens is called changing the reading frame. So essentially what that means is all of the codons get shifted down one. So the original codon in this particular protein was CGU. But now because we've added a base, now the reading frame is UCG. So you see how everything got shifted down. Well, that shift is called a frame shift mutation. And so that'll come up in just a second. But every amino acid after that particular mutation is affected. So all of these amino acids are incorrect. And a deletion mutation is kind of what it sounds like as well, and also a frame shift mutation because it again changes how the codons get read. So there should have been a G in the DNA here, which would have caused a C to get transcribed. So the original codon again was CGU, but because we deleted a base, that got changed now. So it's the whole mRNA got shifted, so it's GUA. So a frame shift mutation just is one that's it changes the way that the mRNA is read because of the insertion or the deletion of one of the bases. Chromosomal mutations are different because they're changing whole sections of chromosomes, and it affects the entire chromosome. And so it can affect many, many genes on that chromosome. So we've got an original chromosome here that's color-coded in its A, B, C, D, E, and F. And so we just labeled chunks of the chromosome, and each chunk has several genes. So a deletion mutation is one that we saw as a point mutation as well, but the difference between deletion at a chromosomal level versus deletion as just a point mutation is here we're deleting an entire section of a chromosome instead of just deleting one individual nitrogen base. Duplication is where you have a whole extra copy of a portion of a chromosome. Inversion just means to switch or change the order of. And so in an inversion mutation where we started out with A, B, C, D, E, F, we change the order of B and C. So the order of genes on a chromosome makes a difference in terms of making correct proteins. Translocation mutations are probably the most difficult to understand because here we've taken this G and H, it's from a different chromosome. And it's not from a homologous chromosome like crossing over, it's from a totally different chromosome. And so this totally different chromosome has a piece that breaks off that can fit anywhere into this chromosome. So here we've added G and H, just inserts itself in between B and C to create a chromosomal mutation. So a lot of mutations are actually neutral, and the reason is because the genetic code is redundant. So if you look at that big wheel that we used to transcribe, or actually that we used to translate our proteins, you'll notice that sometimes 4 up to 6 different codons stand for the same amino acid. So if the last letter in the codon gets changed, sometimes that doesn't matter at all. It's still going to make the same protein, and so that mutation is considered neutral. A lot of the mutations that we hear about are bad because they're caused by diseases, or they cause diseases rather, and those diseases can be passed down from parent to child. However, some mutations are a good thing. Mutations can increase genetic diversity, and that's good because it provides variety. It might provide a beneficial protein or a helpful protein that could help out an organism. It can also be a good mutation in plants. So sometimes what happens in plants is there's a mutation that causes a problem during meiosis so that the two sets of chromosomes that usually separate, it causes them to not separate. So then you end up getting gametes that have two sets of chromosomes instead of just one. And so those gametes come together and fertilize, and they grow into a plant. And so now this plant, every cell in this plant has 4 copies of each chromosome. So actually, this is a good thing for food. So having more than 2 sets of chromosomes is called polyploidy. So poly means many, and ploid has to do with sets of chromosomes, kind of like haploid, diploid. Polyploid means many sets of chromosomes. So polyploidy actually causes plants to be larger and have hardier fruit. So hardier just means that they can survive more conditions like cold weather and drought. So most of the strawberries that you buy at the grocery store actually have 16 copies of all their chromosomes.